Good morning. Good morning, students. Am I audible?
Good morning, ma'am.
Okay, that is
Good
fine.
afternoon, ma'am.
Good afternoon, ma'am.
Good afternoon. Good afternoon. Okay, so audible. Right. So today's lecture is a repetition because viruses you've already done a lot of things last semester, but still I thought it will be a revision, right? So I thought we should go for it. And before that, last time we discussed bacteria, right? So quickly just revise what are the symptoms which are shown by the bacterial pathogens in the plant. Yes, who will tell me? Except Manaswani. आप लोग पढ़ते नहीं हो चलो मैं नाम लेना स्टार्ट कर दूं चलो अधीश बताओ कौन सा सिम्टम होता है वायरस का एक एक सिम्टम बताना स्टार्ट करो सब Shivani don't raise a hand I'll ask myself now because when raise hand only few people there's a few people or a few girls I must say that boys in the class are sleeping right now so only few girls they answer the questions my questions so it's better to take name and ask question right yes Adhish name one symptom caused by bacteria in plant hurry up unmute yourself
Yeah, yes, ma'am. So,
Mhm. Yes, any one symptom? I did not hear you, Adhish.
uh, ma'am, bacterial symptom.
Yes, bacterial symptoms in the plant.
Ma'am, leaf curling, yellow ring spots.
Okay. Yes, Ayushi, another.
Ma'am, leaves, spots, blights, wilts,
Sam,
scabs.
Devanshi.
Ma'am, leaf spots or wilts.
Liz, yes. Ishita.
Ma'am, leaf curls, stunted growth in plants.
Janvi.
Ma'am, leaves, spots, wilts, and scabs.
Mm-hmm. And students, just give me 1 second. I have a call from the office. Just 1 second. Okay, sorry for that. There was a call from the office. So, okay, हम बात कर रहे थे बैक्टीरियल सिम्टम्स की. ठीक है, हम कहाँ पहुंचे थे? इशिता के पास पहुंचे थे शायद. इशिता, आपने आंसर किया था?
मैम जानवी तक हो गया
Yes,
था.
ma'am.
जान तक हो गया. चलो क्षितिज. क्षितिज, टेल मी वन सिम्टम कॉज्ड बाय बैक्टीरिया इन प्लांट. क्षितिज, मैं तुम्हें अटेंडेंस नहीं दूंगी आज अगर तुम नहीं बोले. You never answer question in the class. When I answer question, आप कभी unmute नहीं करते हो.
Can you repeat?
Tell me one symptom which is caused by bacteria in plant. Just name one, any one symptom. लास्ट क्लास में याद है हमने बैक्टीरिया पढ़ा था और उसके कुछ सिम्टम्स भी पढ़े थे. आई एम वेटिंग क्षितिज. क्षितिज, आई एम स्टिल वेटिंग.
अभी याद नहीं आ रहा.
क्यों नहीं याद आ रहा? फंगस के सिम्टम बताओ क्या होते हैं. सिम्टम क्या होता है? टेल मी व्हाट इज सिम्टम. क्षितिज, टेल मी व्हाट इज सिम्टम. व्हाट डू यू मीन बाय सिम्टम?
Which is जो कैरेक्टर जो प्लांट में बेसिकली शो होते हैं, जैसे फॉर जैसे जैसे कभी रेडिश ब्राउन लीफ रहते हैं, तो बेसिकली तो जो प्लांट है वो
हाँ.
पॉइजन.
जो सिम्टम्स जो विजिबल चेंजेस प्लांट में आते हैं जब कोई इन्फेक्शन हो. अब बताओ कौन कौन से चेंजेस दिखते हैं तुम्हें? कौन कौन से सिम्टम्स मैंने बताए थे? फंगस और बैक्टीरिया के. वायरस के बताओ. चलो, वायरस तो पिछले साल पढ़ा है ना.
Yes,
हाँ, बताओ एक सिम्टम का बताओ. आई एम नॉट गोइंग टू लीव यू अंटिल यू आंसर. वायरस का नाम बताओ. एक किसी वायरस का नाम बताओ. प्लांट वायरस. टोबैको पे कोई वायरस होता है ना जो सबसे पहले आइडेंटिफाई हुआ था?
TMV, टोबैको
हाँ.
मसाका.
तो मोजाइक इज द सिम्टम, राइट? Start studying, Kshitij. You should study more, right? Yes, after Kshitij, who's there? Manas, Sunny, okay, leave Manas, Sunny. Namrita, also leave. Nehal?
Yes, ma'am.
Yes, any other symptom which is not mentioned by other student?
Ma'am, fungal or bacterial?
Bacterial.
Ma'am, bacterial. Ma'am, canker or wilts.
Yes,
And
yes, cankers. बस रुको वहीं पे. रूपल?
Yes, ma'am.
Yes. Any
Ma'am,
symptom
it
caused by
it
bacteria?
can be leak spot, necrosis.
Necrosis. बस रुक जाओ. साहिबा?
Yes, ma'am.
Yes, Sahiba.
Ma'am, soft roots, soft rot
Yes.
of roots.
Soft rot. Yes. Good. Sanjana?
Ma'am, wilts, scabs.
Scabs. Shakti?
Ma'am, scabs, cankers, and overgrowth.
ओवरग्रोथ को क्या बोलते हैं? कुछ टर्म यूज की थी हमने ओवरग्रोथ के लिए. शिवानी बताओ तुम.
Ma'am, necrotic lesions, wilts,
नेक्रोटिक लीजन्स
cankers.
ओवरग्रोथ नहीं होती है. क्राउन गॉल सुना था किसी ने? हमने बताया था कल. एग्रोबैक्टीरियम tumefaciens, they cause galls basically, right? So galls are
Yes,
the
ma'am.
basically overgrowth which are being caused by, right? Fine. So there are number of we discussed about number of the symptoms which are being caused by the see in bacteria. I said this yesterday also that bacterial and fungal symptoms are similar. Like scabs are caused by bacteria also, fungus also. Necrotic lesions are caused by both. Blights are caused by both. Wilts are caused by both, right? So similar symptom, fungus and bacteria, they usually show the similar kind of symptoms, right? So today's lecture is total a repetition of viruses. So We'll just quickly go through this. We'll revise what are the symptoms. I guess you all know this. So we know the virus is very small, so small that it is called as an ultra microscopic, that we cannot see through the microscope. We need an electron microscope. It is around 20 to 300 nanometers in diameter, right? So very small, composed of RNA or a DNA or a protein coat, and sometimes an envelope also. Some viruses are enveloped viruses also, right? So it is very small size, and it can cause disease in plants, animals, and bacteria. And the word virus means slimy liquid or a poison, right? So again, they are intracellular obligate parasites. They are not cells, but they are infectious agents that contains protein coat and nucleic acids. They can infect all of us, right? They can infect even the viruses or even other pathogenic organisms, such as they can infect bacteria also, right? So they are infectious. They cause the number of diseases in plants and humans. So comparison, if we talk, they are virus. If we compare virus and fungi, so virus are the smallest. Fungi are spore producing. We can still see the spores. Definitely, we cannot see the ultrastructure of a spore, but a black powdery mass sometimes can be seen on the plant. We can see that thing, right? Then the virus, they have a DNA, RNA as a their genomes. They encode some proteins such as core protein, movement protein, or a replication protein. Fungi, they have structures such as mycelium, hyphae as part of their infection mechanism. Then how do viruses differ from bacteria? So bacteria are a living unicellular. Viruses we still don't consider living. They don't have cells, right? But they both can cause diseases, right? So bacteria are larger, viruses are smaller. Bacteria, they reproduce by fission. Viruses, they need some living host for the reproduction and multiplication also. Bacteria can be treated with antibiotics. We cannot treat viruses with an antibiotic, right? So there are many differences. So you should know the differences between fungus, bacteria, and viruses, and similarities, right? This is important. So there are then plant viruses and animal viruses both. So definitely a plant virus infect plant, animal virus infect animal, and we are lucky that they cannot cross their borders, right? So animal viruses cannot infect plant, and similarly plant viruses cannot infect animals or humans, though animal viruses can infect humans. So those diseases are called zoonotic diseases or zoonotic viruses, which can transfer from animals or birds to the human, right? But plant diseases or plant pathogens, they do not impact us. We even if we consume the fruits or vegetables which are infected by viruses, we don't get any harm from them, right? So it can be, it is easy, it is okay, fine, we can have them, right? Then genome, plant are mostly RNA, whereas human or animal are mostly DNA. And examples, there are number of tobacco mosaic virus, potato virus Y, cowpea mosaic virus, number of thousands of examples. And human, we have a latest coronavirus pandemic, which we still are facing. I think it started in early 2019, somewhere in December, and we have entered 22. We are still impacted by this virus, and we are still under the pandemic right now. So let's hope this is the last wave. Number of diseases are being caused, and plant viruses, they enter to the host cell through wounds or through holes or through vectors, basically. And human viruses through the endocytosis, they enter the human, right? So viruses, these are the definitions we know. Genome, yes, we know RNA virus. Genome could be your DNA or RNA, right? Shape could be linear, circular, or segmented. They could be single-stranded or double-stranded. They could be positive sense. They could be negative sense also, right? So most viruses, RNA genomes, and some with single-stranded DNA genomes, they are either positive sense called plus strand or negative sense called minus sense. So this depend upon their complementary. They are to the viral messenger RNA, right? So positive sense viral RNA, they are usually in the same sense as the viral mRNA is there. So they can be translated by the host cell, whereas negative sense viral RNA is complementary to mRNA, right? So they are not in the same sense. So they must be first converted to positive sense RNA by using RNA-dependent RNA polymerase before the translation takes place. So a single infectious virus particle is known as a virion, a single particle that exhibits ability to infect a host, right? So when we know a virion, virion means a virus particle which is capable of causing infection. A protein coat that covers the nucleic acid is called a capsid. And some viruses, they have their capsid which is surrounded by a lipid-like coat which is called as an envelope also. And the capsids or a coat protein are basically being exploited in many ways. They have many important properties. They are antigenic, right? So when we are talking about a generation of raising of antisera, so we can just use coat protein genes. We don't need to use whole virus, and we can develop antibodies against that particular virus, right? Then shapes, there are several shapes, rod shape, polyhedral, or helical shapes also, right? Enveloped viruses also, so there are a number of shapes. Isometric viruses, twin virus particles, which are called Gemini. So number of shapes are there, right? Then, okay, symptoms. So again, symptom is the visual changes which are being produced by any pathogen, right? So these are important to study a particular disease, and they play a very important role in the identification or diagnostics or diagnosis also, right? So we know in viruses, it is very easy to know the symptom because the name of the symptom is always included with the type of virus or a disease, right? For example, we called mosaic diseases, right? Yellow diseases. So it's very common that mosaic diseases means mosaic symptom is the common, yellow diseases means yellow disease, yellowing of the leaf is the common symptom, right? Similarly, the symptom is when we talk about the nomenclature of plant viruses, especially the symptom is there in the name, right? Tobacco mosaic virus, tomato leaf curl virus. We have apple chlorotic leaf spot virus, prunus necrotic ring spot virus, cucumber mosaic virus. So in all the viruses name, you'll see that there is one major symptom which is being shown by that virus, which is included in the name of the virus. So it is very easy to identify or guess the, or it is not the only symptom that is being shown by that virus, but it's the main symptom which is being shown. Besides that, there could be other symptoms also which can be shown by that virus, right? So virus infection also not very important that all the time it can cause disease in all the infected plant. So there are certain conditions where sometimes disease could be absent, right? For example, if infection is with a very mild strain of the virus, then will not see symptom. If the host is tolerant, then also will not see symptoms. Or if it is recovery from disease symptom in a newly formed leaves, so in those leaves also will not see symptoms, right? Some leaves, they escape infection because of their age and position on the plant, so those leaves are also free from the symptom. Then plants infected with cryptic virus. Cryptic ka matlab hidden viruses. Sometimes these viruses, they are present always in the plant, but not major symptoms are being shown on the plant, or the plants are symptomless, right? So in those cases, we don't see the symptom, but still virus could be there, right? So loss, again, we don't, we know this, right? Why we need to study viruses, a lot of losses are being caused. Then some type of a direct and indirect damage are reduction in a growth, crop failure, or reduction in a market value, defects of a visual attraction, size, shape, and color. And okay, symptoms. So this is important, right? So symptoms, macroscopic and microscopic. Macroscopic means those are visible by a naked eyes to us. Microscopics are usually that we see under the microscope the symptoms which are being caused, histological changes in the plant, right? So macroscopic means local symptoms, right? So localized lesion which develops near the site of entry on the leaves, right? So ज़्यादातर ये biological assays may we use this for the identification of a plant pathogen, viruses especially. So they just at the site of the entry, some necrotic lesions are being shown on the plant that are called local symptoms, right? Then we have a systemic symptom. So systemic symptoms are the symptoms which are shown throughout the plant. It could be the root also, fruit also, stem also, and the leaf also, right? So it affects on the size of the plant. So reduction in a plant size is a most common symptom induced by virus infection. So overall stunting of the plant is seen, right? Then stunting may affect all parts of the plant, leaves, flower, or a fruit. So this image shows stunting caused by tomato bushy stunt virus. So it reduces the fruit size also, and there are a misshapen fruit also. They are not of a good quality fruit which are being produced, right? Then we have a mosaic patterns and a related symptom. So this is a very common symptom shown by the viruses of the plant. So in development, this what does mosaic means? It's a pattern of a light and a dark green areas, giving a mosaic effect on the infected leaves, right? So again, some dicot plants, the mosaic are generally irregular in this. These are to differentiate basically. So they have noticed that in dicot, the mosaic pattern is irregular, or there may be only two shades of a color, dark green, pale yellow, or yellow green, right? Examples here are given about we can see the African cassava mosaic virus and evolution mosaic symptoms. So similarly, we have a tomato mosaic virus, we have a cucumber mosaic virus. So a lot of mosaic symptoms are shown in the plant viruses. Again, they may be different shades of green and yellow, as with the turnip yellow mosaic virus in Chinese cabbage, right? So there are number of mosaic type of a pattern which are being shown here. Again, there's a variegation or a breaking in the color of the pattern, which is called color breaking also, right? So this is commonly it accompanies mosaic or a streak symptoms in the leaf. The breaking usually consists of a flex, streaks, or a sectors of a tissue with color different from the normal. So color breaking जब आपको flowers में दिखता है तो it shows patches, right? So this first example is the tulip color breaking virus is the very important virus, and instead of causing loss to that particular crop, it increased or it enhanced the beautification of that flower, right? So flowers, tulip flowers with the variegated colors, that means the two colors, white and pink, white and yellow. So they were more in demand, right? So that was the positive effect shown there. But in some flowers, yes, it do show some negative impact also, that they don't show a very good single color if single color flowers are being required, right? So flower breaking or a color breaking is again a very important symptom shown by the viruses. Then we have yellows diseases. So named is indicate yellows. That means yellowing of the leaves or a plant. Could be due to the virus infection. So viruses that cause a general yellowing of the leaves are not as numerous, those causing mosaic diseases. But some viruses causing yellows in sugar beet, they are important. The first sign of infection is usually clearing or yellowing of the veins. So we know that flowers, leaves, we see there are number of veins in different pattern. We'll see the veins. So the yellowing of the veins in the younger leaves is the first symptom, and then it could be slight or a severe. No mosaic is produced, but in some leaves there may be sectors of yellowed and normal tissue also, right? In strawberry yellow edge disease, yellowing is largely confined to the margin of the leaf. So different kind of a yellowing symptoms are also being seen. Then leaf rolling, virus infection can result in the rolling of the leaves, which is usually downwards. Very occasionally it's onwards towards the plant, but usually it's downwards. So this is example of a grapevine leaf roll virus. Then a marked symptom in many virus diseases is a pattern of concentric rings and irregular lines on the leaves and sometimes on the fruit also, right? So these lines may consist of a yellow tissue, could be the death of the superficial layer of the cells, giving a itched appearance. And in some cases, complete necrosis through the full thickness of the leaf lamina may also occur. So this is a papaya ring spot virus, and the second one is a prunus necrotic ring spot virus on the leaf. So we can see necrotic spots on the leaves. Papaya ring spot virus, these are the initial symptoms, right? So after some times, these spots will become necrotic and will be brown or some other dark color. This is again papaya ring spot virus. You can see the spots. Then necrotic diseases, death of the tissues or whole plant is the main feature of these kind of diseases. So necrotic pattern may follow the veins as virus moves into the leaves, and sometimes the whole leaf is killed. Necrosis, again, it can extend throughout the plant. For example, in potato virus X and potato virus Y, necrotic streaks, they also appear in the stems also, right? So necrosis spreads rapidly to the growing point, which is killed, and subsequently all leaves may collapse and die. So necrosis result in the death of the whole leaves. So wilting of the parts that are about to become necrotic often precedes such systemic necrotic diseases. So after that, wilting of the plant also take place. Then we have a pox-like symptom. See, you can see the pox-like symptom. This is a pox-like symptom in a peach, I guess, right? Then a bending of the vein. So you can see there are veins in the leaf, right? And you can see bending is that when there are bands of yellow color in those leaves being shown. So this is called vein bending. This tomato spotted wilt virus in tomato we can see, and on the fruit you can see these kind of rings in case of a tospovirus, right? So these are the symptoms. Then we have a tobacco leaf curling. Different kind of a leaf curling symptoms are being shown by the viruses. Could be tobacco leaf curling and mosaic also combined by mosaic. Then these are the Begmovirus symptoms you can see, right? Different kind of a symptoms. Somewhere we see the vein clearing, somewhere there's a vein bending. This is a puckering of the leaves and mosaic. So puckering is leaf, they become little puffy kind of a structure is being shown in the leaves, right? The leaf curling is also there. So different kind of a symptom shown by the Begmoviruses, which are DNA viruses affecting plant, right? Then this, I guess, we have discussed many times that what are nutrient deficiency symptoms and how they are very similar to virus symptoms, right? So we can see the different deficiencies can lead to the yellowing of the leaves, or sometimes some leaves become pain, whereas veins are green. So these kind of symptoms can confuse us for the viral symptoms also. But then I told you that one common identification is that if there is a nutrient deficiency in a particular field, the chances are that whole plant or all the plant in the field, they'll show some common symptoms, right? Whereas if there's a virus infection, then definitely not all plants are going to show those symptoms. Only few plants in a patches you'll see the symptoms. So this is how we differentiate the symptoms on the field. Then here, see nitrogen deficiency. Again, the nitrogen deficiency in the corn. So these are the again nutrient deficiency symptoms. This is again yellow mosaic disease of a mung bean. So see, in whole field, only one patch is showing the symptom, not all mung bean crop is showing the symptom. This is how we differentiate here, right? Then mung bean yellow mosaic disease here. See clearly the yellow and mosaic on the leaves. They are very clearly seen in few leaves. Then again weeds, we say symptoms on the weeds also because they play a role in a very important role in a plant virus transmission. So this is a Geratum weed. It is a very common weed which is found in cities, anywhere, any open space near the field also. Everywhere you'll find the Geratum, and this will show these yellow kind of a symptom, which indicates that they have a certain kind of a viruses infection on them, right? This is again, croton is also a weed. This weed is a very common, this weed is, I've seen this weed throughout in Delhi NCR. In my locality, I see this weed growing, and but I'll see the change in symptoms, right? During the winter season, I'll see less symptoms. During the rainy season, I'll see more and more symptoms, right? So this is how with season also the viruses impact is varying, right? So croton, again, it's a very common weed, and a lot of viruses are being identified on this plant. Then there are microscopic or histological changes. In affected plants, so the common one is the inclusion bodies where viruses can be identified. So virus infection leads to the formation of inclusion bodies. So if we see it under the microscope, we can see those inclusion bodies, right? So necrosis is the death of the host cell is called necrosis. Streaks and spots, they are necrotic spots. Then there's a hyperplasia and hypertrophy. So excessive growth of host tissue is caused due to hyperplasia and hypertrophy, right? So in hyperplasia, the overgrowth is due to increased, there are two different terms, hyperplasia and hypertrophy. Both cause excessive growth, right? But there's a difference. Hyperplasia, the overgrowth is due to increased cell division and cell number constant. Then in hypertrophy, the overgrowth of organ is due to increase in size of the individual. So you see the difference. In one, the cell division is taking place, and in other one, the increase in size is the individual cell is taking place, such as witch's broom of cherry or a corn gauze, right? All those things. Then cytological changes, a virus infection infect various cell organelles like nucleus, chloroplast, and mitochondria. Nucleus induces formation of nuclear inclusion bodies which affect the nucleolus also. Nucleus, they become granular, chromatin may be reduced, disintegrated. Just give me 1 second. Okay, so chloroplast also show disintegration, swelling, or clumping. Mitochondria show aggregation in virus-infected cells or formation of vesicles. Then cell walls also show thickenings or a deposition of electron-dense material between cell wall and plasma membrane or a callus deposition. So these are the cytological changes which are being seen. Inclusion bodies. So these are intracellular structure which are formed due to the virus infection. May contain virus particles or a virus-related material. They can be a nuclear or a cytoplasmic. That means they are found in nucleus also and in a cytoplasma also. They may be amorphous, crystalline, or a pinwheel, different shapes and structures. Inclusions are found in the epidermis in case of a tobacco mosaic virus. They are found in the vascular tissue in cystus disease virus, and in the nucleus they are found in case of a Gemini virus and Hebdovirus. So what does this mean? Why this is important? So on the basis of their location and their shape, we can differentiate the viruses also, or the genre of the viruses also, that if they are found in the vascular tissue, it indicates the citrus disease virus. If they are found in the nucleus, it indicates the Gemini viruses, right? So they can be used as an identification tool here, right? These are the crystalline bodies. They are form of a virus aggregates which give a three-dimensional like appearance when viewed under the microscope. They accumulate in the cell cytoplasm. They are mostly seen in epidermal and hair cells in the cytoplasm. They could be in the form of a rods, helical, icosahedral, or a crystalline arrangement of the infected cells. Then we have a pinwheel inclusion. So as the name suggests, they form pinwheels, right? Wheel-like a structure. Pin, you can see the dotted structure. They are called as a pin and a wheel-like a structure. They are important characteristic feature of a Portiviridae family of viruses. They are very commonly seen in a Protovirus Y, bean common mosaic virus, and a soybean mosaic virus. So if we see a pinwheel inclusions under the microscope, so it indicates a particular family where it is being found. Then nuclear inclusions are considered to be a main characteristic feature of a Gemini viruses, right? So these again are aggregates of a virus particle, because we know the DNA, and Gemini virus is a DNA virus, right? The inclusions stain bluish to purple when we stain it with a particular stain, right? So blue stained nuclear inclusions in the phloem tissue of an infected plant can help us to diagnose a Gemini virus infection. So that's why I told you that these, since there are different types of inclusion bodies which are being found in a different kind of a plant cells, right? So on that basis, we can identify them, that which virus infection is in a particular plant on the basis of the location of this inclusion body that can be done, right? So this is all for the virus infection or virus symptoms on the plant. So with this, we finish the major viruses, not viruses, the major pathogens which infect plant, in which we have studied fungi, we have studied bacteria, and we have just studied the viruses also, right? So the major symptoms and the major pathogens that we have done, right? We have nematodes also, if I'm not wrong. So we'll pick up that also, maybe one lecture. But the major pathogens are done, right? So this part is very important, right? Because we know here we know the basics of a particular pathogen, that what are those pathogens, and what kind of a symptoms they are causing on the plant. So this is very important, right? So the I guess we are done with the symptomatology part, and about nematodes I'll discuss in detail with the diseases that I'll discuss of those pathogens, not now, right? So after this, the other lecture that I'm going to take is about the history. First module we are doing right now. So the another topic in the first module is basically the history, I guess, right? Let me just go through that. Yeah. The first module was the definitions, okay, symptoms, types of plant pathogen, then contributions of important plant pathology. So that would be the last topic of first module. That means history or a major landstones or a major plant pathologist who have contributed in the past, right? So that will be the history part that we are left with the first module, right? So yes, if there's any question or anything else which you have not understood, please ask your questions. Open for discussion, right? And in the meanwhile, this time I'm going to take an oral discussion part, not a written question, right? Okay, fine. No question then, right? So just let me, since viruses, I guess you एक पूरा course आपने virus पे पढ़ा है, तो I'm sure you know a lot about viruses, right? Okay. So who will tell me how do we diagnose viruses? अभी अभी एग्जाम दिया ना आप लोगों ने. How do we diagnose viruses? Just name the method.
You can by using serological methods or molecular methods.
Yes, by
Like
serological. Yes,
using
stop
PCR.
there. Stop there. Stop there. I'll ask someone else. Yuvraj, stop. Yes. Yes. I will ask. Yes, Nandini, can you tell me what are the different serological methods that we use, or just one? Okay. Serological method that we use for virus identification. Nandini, can you hear me? No. चलो रूपल तुम ही बता दो. Yes,
Ma'am,
Rupal.
ma'am, we can use PCR,
I
ELISA.
asked serological. Which is serological? Which is serological method? PCR और ELISA में से serological कौन सा है? मुझे ये बताओ चलो. अब ये मत मुझे वो करना कि तुम्हें ये नहीं पता serological कौन सा और molecular कौन सा. What does serological means? What is serology? ये शिवानी, आपने लास्ट टाइम इम्यूनोलॉजी भी पढ़ा है और वायरोलॉजी भी पढ़ा है, तो सेरोलॉजी आपने पढ़ा है अच्छे से. आई नो. व्हाट इज सेरोलॉजी? हाँ शिवानी बताओ.
Ma'am, diagnosis of the blood serum, like
Yes.
diagnostic methods.
Diagnostic method which uses a blood serum, serology, right? So ELISA कौन सा हुआ? Serological हुआ या molecular हुआ?
Ma'am,
क्या use
ELISA is serological.
Yes, ELISA is a serological, right? So what? Yes, the next one. What are molecular methods? हाँ दिवांशी बताओ. Devanshi.
Ma'am, am I audible?
Yes, Devanshi, you are
Ma'am,
very much audible.
molecular methods can be PCR.
Yes, molecular methods में सबसे important PCR है, जिसमें आपका RT-PCR है, उसमें आपका real time PCR भी है, right?
Yes.
There are so many methods, right? So diagnostic methods are serological and molecular both, right? Okay, fine. So who will tell me about the common symptoms which are caused by viruses? भूलोगे नहीं तुम symptoms, मैं इतने अच्छे से तुम्हें याद करवा दूंगी सबको. Yes, Ishita. Yes, Ishita.
Ma'am, am I audible?
Yes.
Ma'am, common symptoms of viral infection are mainly headache, body pain, body pain, and in some cases rashes will be appear.
Okay, fine. It's my fault. I should have asked virus plant symptoms. Anyways. Okay, virus plant symptoms मुझे कौन बताएगा? युवराज बताएगा. युवराज, चलो बताओ.
Ma'am, hormonal imbalance, stunt growth, nutrition deficiency.
न्यूट्रिशन डेफिशिएंसी अलग सिम्टम्स है बच्चा, वो तो मैं इसलिए कंपेयर करके बता दी हूँ कि न्यूट्रिशन डेफिशिएंसी सिम्टम्स सिमिलर होते हैं वायरस की तरह, है ना? सो न्यूट्रिशन डेफिशिएंसी आर नॉट द सिम्टम्स कॉज्ड बाय द वायरसेस, दे आर
Okay,
द
ma'am.
सिमिलर
And
सिम्टम्स, है ना?
yes, ma'am.
डोंट
Then
कंफ्यूज दैट, राइट?
mosaics and leaf curling.
Yes, mosaics, leaf curling, yellows, stunting, lot of symptoms are being caused by the viruses, right? Yes, good. I can see so many hands raised up, right? At least virology अच्छे से पढ़ी है आपने, so this gives me good feeling. मैंने कुछ पढ़ाया है, right? So when you guys don't answer about the course that I have already taught you, और इतने अच्छे से मतलब plant pathology is my specialization, and I have been working on plant viruses since a very longer time. So that is the subject where I specialize. So when you teach a subject which is your specialization, so you teach it differently than the other subjects where it is not your specialization, right? So viruses, yes, they are my specialization, especially plant viruses. So I've read a lot about animal and human viruses also, but plant viruses definitely somewhere, somewhere in 2003, I started working on plant viruses, and it is coming a 2023 very soon. So a lot of time I have spent on that, right? So when I teach in a, when you're teaching with your whole heart, then you expect students also to give their best, right? So that is the only thing. So what I want is that whatever I am teaching you, and I'm repeating so many times, so many things, so that it goes into your brain, right? To remember those things that are being taught in the class. So there's a reason for that, so that you remember that, right? So some after graduation, you all will be going somewhere else, and whenever some these kind of a basic questions will be asked, so you'll be able to answer. So that is the good thing, that, right? Because you've studied plant pathology and even a virology course. So this is important, because in graduation usually we don't study these hardcore courses, especially the virology one. So this is an important subject, right? So just and since we see what viruses can do, we all are witnessing that. So they are very important as a pathogen, right? Okay, thank you so much, students. So I'll take your attendance and I'll take your leave. Adhish Mishra,
Present, ma'am.
Ayushi Jain,
Present, ma'am.
Sam,
Present, ma'am.
Devanshi,
Present, ma'am.
Ishita,
Present, ma'am.
Janvi Singh,
Present, ma'am.
क्षितिज.
Present, ma'am.
क्षितिज अगली क्लास में पढ़ के आना, तुमसे मैं जरूर हर क्लास में क्वेश्चन पूछने वाली हूँ अब, तो मेरी हर क्लास में पढ़ के आओगे तुम, अब बता दिया मैंने. मानसवनी,
Present, ma'am.
मानसवनी के बाद नम्रता,
Present, ma'am.
नंदिनी, नेहल, रूपल,
Present, ma'am.
Present,
साहिब
ma'am.
या साहिबा, संजना,
Present, ma'am.
शक्ति, शिवानी,
Present, ma'am.
तन्वी,
Present, ma'am.
त्रिदेव,
Present, ma'am.
विवेक, युक्ता and युवराज, right?
Present, ma'am.
Present, ma'am.
Okay. So this time I think your workload is not that much. I see very less classes because you have a project also in the end, right? So you guys are a lot of time, you guys are free, okay? Isn't it?
Ma'am, we have to do our NGO work also for human value course.
वो कोर्स
So-
आपको ना पिछले सेमेस्टर में लेना चाहिए था, I guess. That was you should have taken in that course in earlier
वो
semester.
तब available नहीं था.
Available nahi tha, okay, because it's easy, because this time you're stuck, you'll be doing 1 month project also, and simultaneously they would like you go to the NGO. At that time it will not be possible. I don't know how you guys are going to do that, because I really hope that this time you do project in some lab, it is not online thing, because in online we don't learn that much things, right? So we really hope that this peak in the Feb get over, and by the time it's your project time in the March, you're able to do your project somewhere outside, right? And also, since you have a free time, so utilize that time, because it is the time that you should start thinking what exactly you're going to do after your graduation, right? So utilize this time in thinking that and collecting information and looking for the various things. वो graduation करने के बाद नहीं सोचना है, पहले सोचना है कि graduation के बाद क्या करना है, right? So that you don't waste your time. आपके time waste now. Time is very important. Okay, so just start looking that also, and just keep asking your mentors and faculties about the suggestions and the help, so that they can guide you what exactly or what all the are the options you can look forward for, instead of doing it कि अब मैं graduation करके and then I'll think what next. No, this is your last semester. This is the time that what you start thinking what should be your the next step now, right now. Okay, so do that also. Fine, I'll see you tomorrow in the practical class. Yes, Manaswini, you want to say something?
Do you know when our 5th semester marks are coming out? Because I have to apply to
It
college.
should, I feel, Manaswini, since university is closed, you know, from this week onward, we are also working from home. So there could be certain delay, but I believe that all exams are checked and evaluation is done. So anytime you can expect your result, because evaluation is done long back. They must be collecting those internal marks or practical marks that are also submitted. So I think very soon you are going to get. वैसे 10th January थी deadline, but due to this COVID, I guess थोड़ा सा हुआ होगा, but maybe anytime soon you're getting the result, I'm sure, right?
Okay, ma'am. Thank you.
Yeah. Okay, students. So I'll see you tomorrow in the practical class. Bye bye.
Thank you, ma'am.
Thank
क्या
you,
हो गया
ma'am.
देवांशी, तुम्हें इतनी
Thank you.
बहुत ही ऐसी थकी हुई थैंक यू बोली है ना, ऐसा लग रहा है मैंने पता नहीं कितना थका दिया है तुम्हें.
No, ma'am, actually, my wisdom tooth is causing me a lot of pain.
ओहो, मतलब तुम्हें विजडम अब आई है. गुड, कंग्रेचुलेशंस फाइनली.
No, ma'am, it's my second one.
अच्छा, सेकंड विजडम आई है, ठीक है. ओके, आई विल सी यू टुमारो स्टूडेंट्स. बाय बाय.
Thank you, ma'am.